I'm going to be talking about something a bit different. When we look at the hallmarks of aging, and you have already seen this slide a couple of times, there is one that our lab is particularly interested, that is this disabled autophagy. Most of you know that autophagy refers at the way in which cells clean any kind of damage from inside the cell, but also utilize it to recycle. So when you have an organelle or a protein that needs to be eliminated, the lysosomes will degrade it, and then the byproducts, the amino acids, the lipids, the sugars coming from that degradation will be utilized again from the cell. However, as we get old, we and others have shown that this cleaning system doesn't work anymore, and then you start accumulating unwanted products inside the cell, and you also have problems with recycling. So I'm going to be talking mostly about autophagy, and there are different ways in which you can deliver this garbage into the lysosomes. For example, if this is your lysosome, you can just trap what you need to be degrading inside vesicles that then fuse with the lysosomes and result in degradation. And this process is known as macroautophagy and comes in different flavors depending on what you are degrading. For example, lipids, you talk about lipophagy; mitochondria, you talk about mitophagy. But this is not the only way in which the cells can do autophagy. In mammals, you can also invaginate the membrane of the lysosome or the late endosome and trap materials of the cytosol in what is known as microautophagy. And then there is also the possibility of getting single proteins and make them cross the lysosomal membrane for degradation. And this process is known as chaperone-mediated autophagy, and I will be mostly talking about that process today. So I want to convince you at the end of the talk that chaperone-mediated autophagy, that I will be referring as CMA, is a potential gerotherapeutic targeting. So the process is very simple. Basically, you have a single protein that contains a targeting motif, and it's going to be identified by a chaperone that brings it to the surface of the lysosomes. And the only protein that you need to remember is this lysosomal associated membrane protein, type 2A, that is the receptor that becomes also a translocation complex for the proteins to go inside and get degraded. CMA is constitutively active in all the cells in your body as far as they have lysosomes, but is maximally activated in response to a variety of stressors, as you see there. CMA have different functions. So one of them is quality control, removing damaged proteins. Also replenish of amino acids, right? When you degrade a protein, those amino acids can be now reutilized for energy and for synthesis. But also because CMA can degrade fully functional proteins to terminate whatever they are doing in the cells, CMA also has important regulatory functions in the control of metabolism, cell cycle, cell differentiation, activation, and the list keeps growing. So alterations in CMA has been associated with multiple disorders. You see here neurodegenerative conditions, eye disorders, metabolic, cardiovascular, cancer. And the major commonality of all those diseases is that, as we know, there are diseases of aging. So we've been very interested in trying to elucidate how this pathway changes with age and whether it can contribute to the aggravation of some of these age-related diseases. So to study that process, recently we have developed a fluorescent reporter that basically is a fluorescent protein that when delivered to lysosomes by CMA, it will make them glow. So you can count the number of dots, and the more lysosomes are highlighting, that means you have more CMA. And then a very talented former postdoc, Sushan Dang, that now she has her laboratory here in Shanghai, she developed a transgenic mouse model where we can monitor CMA in vivo. So basically this is a slide of the hippocampus of the brain of these animals, and as it gets closer you will see little dots that correspond to activity of CMA. So we can map the activity of CMA in different parts of the organs, but also we can go into single cells, as you see on the right, and see not only that CMA is active, but where this CMA is happening, because you see that the dots occurs in different parts on the cell. So taking advantage of this model, we thought that it was a good opportunity to study CMA changes at the systemic level, and also very related with the previous talk, we were very interested in possible sex differences, because all our studies originally have been done only in males. So taking advantage that everybody in my lab is expert in a different organ, what we decide is to create this cohort of animals, what we analyze CMA in each of the organs that you see there. This work was recently published, so I'm just gonna give you like the bottom line. And something that we found as you look at these UMAPs is that different organs have different basal CMA activity. You have organs like the pancreas or the liver that have very high levels of CMA in basal conditions, and other organs like the brain or the retina, it depends on the cell type, they have different levels of CMA. Then when we compare males and females, so in general, and I know we should not generalize in science, but in general we can see that females have higher CMA activity than males. So for example, if you look in the graphs, this is activity in neurons, in the hippocampus, and as you can see, the males, these are young animals, but the males have significantly lower CMA. But I don't want the males in the audience to get demoralized. There are other organs. For example, if you look at the brown adipose tissue, males have higher basal activity. But in general, for most organs, CMA is higher in females. And the other characteristic is that we found that there is almost a systemic decrease with CMA when you look in aging, but once again, the decrease is more pronounced in males. So if you look again in neurons, in both cases, all animals have lower CMA, but in the males, that decline is higher. And again, there are exceptions. So if you look at the brown adipose tissue, in females, instead of decreasing with age, it turns out that it increases. But having this level of granularity is important because of what we hear. You age in a particular manner, so some organs might age faster, and we think that this might be related with the changes in CMA that you have in part in these organs. So in addition of changes in the total activity of CMA, Rabia in the lab also become interested on whether males and females are degrading the same proteins by this pathway. She was very interested in the central nervous system, and what she did is to analyze proteins degraded in neurons, depending on whether it's neurons from a male or from a female mouse. And we did comparative proteomics, and I'm just showing here proteins degraded by CMA. So males are in the x-axis and females are in the y. And as you see here in pink, there are proteins that are CMA substrates in both males and females. But as you see there, there are some proteins that are only CMA degraded in females and some proteins that are only CMA degraded in males. And this is independent of the total absolute levels of these proteins. So then we did a little of pathway analysis, and it became very interesting the fact that in females, CMA is mostly degrading proteins in the presynaptic compartment in neurons. Meanwhile, in males, it's mostly degrading the postsynaptic proteins in the postsynaptic compartment. We decided to know if this has any kind of functional implications, so we eliminate CMA in neurons in a mouse model, and in collaboration with Pablo Castillo, we start doing recordings. So something that we found is that in both cases, male and females, when you eliminate CMA in neurons, you have an increased hyperexcitability and higher tendency to seizures. But when you look electrophysiologically, I'm just showing here the changes in the presynaptic. So as you see, in the case of the presynaptic signaling, the alteration is mostly in the females, but when you look on the males, on the postsynaptic, it's the males, the ones that have higher hyperexcitability at the postsynaptic level. So those differences on what we are degrading in males and females correlate with the type of functional phenotype that we see. But so far I've been telling you about aging in mice. So does CMA change in humans? Unfortunately, we cannot put yet the reporters in people, but something that we've been doing, and this is work from Mathieu and Adrian, former postdocs in the lab, what they decide is to analyze the whole network of components that modulate CMA, because when you study such a network, you can guess a predicted value for the outcome of this pathway. So basically, we gave weight, how important they are for the network, and directionality, are they activators or inhibitors, and came up with a very simple algorithm to predict CMA activity in humans. And you can do it using RNA-seq, or you can do it using proteomics. So just to show you, when we look, for example, this is published work, and we just predicted how is the CMA changes. So these are healthy individuals, 2 parts of the brain, and as you see, there is a gradual decline in CMA as you age. There are other organs in which this decline is more dramatic. So this is, for example, the aorta, and this is individuals from 20 to 80 years old. They are healthy, they don't have cardiovascular conditions, but independently of that, you see this pronounced decrease in CMA as these individuals get old. You can also use the same indexes that are all published to correlate with disease. So taking advantage of the single cell RNA-seq that was done in patients with Alzheimer, what we did is to calculate the CMA activity in excitatory and inhibitory neurons. And as you can see, the activity decreased with the progression in the stage of the disease, but it's very cell type specific. For example, if you look at the glia, you don't see the decrease that we see in the neurons. And then the beauty is that you can correlate it with pathology. So for example, I'm plotting here predictive CMA activity in the Y against plaque burden in the Alzheimer brains, and you can see that the lower is your CMA activity, the higher is the plaque burden of the pathology. I hope that so far I have convinced you that CMA activity decreases with age, that this is not just an artifact in animals, but it also happens in people, and completely independent of disease, there is going to be a decline on CMA. So then our next question is, what are the consequences? I mean, is this something that we should worry about? And if we think that this is important, can we do something to prevent this decline or to restore normal functionality? So to study the consequences, something that we found is that even there are many reasons why CMA decreases with age. Something that seems very common to all the organs is that that receptor, that LAMP2A protein levels, decrease with age in lysosomes. So when you are young, you have a lot of that receptor, so you can transport a lot of these proteins inside lysosomes. As you get old, the levels decrease. So we decide to analyze what are the consequences of not having CMA independently of the other changes of aging. So what we did is to block CMA in a young animal in a tissue-dependent manner, and this is work from a bunch of people in the lab through the years. But in general, what we have found is that loss of CMA results in degenerative phenotypes. So for example, in the brain, it leads to neurodegeneration, similar in the retina, in the skeletal muscle, also results in a metabolic phenotypes. So for example, you have increase in fatty liver, you have dyslipidemia, the animals develop a full metabolic syndrome. They also have problems of immune senescence. They have reduced increase in each of the, they have reduced increase in each of the stem cell proliferation, and they also have problems with circadian risk and have higher risk of malignant transformation. So based on this, we see that not having CMA phenocopy many aspects of aging. Of course, it's not the only cause, but it's not going to help. So what can we do in order, the other thing we also want to know, so far I have shown you CMA decrease with age, but is this decrease aggravating disease? So Nadia decided to take one of these mouse models that are models of neurodegeneration, and this is the normal course of accumulation of pathology. And if now you impose a decrease in CMA, you see an acceleration of the decrease. And actually, at an age where you don't see A beta in green on the top, you already see A beta accumulation when you don't have CMA. So basically, what I wanted to convey is that not only CMA decrease with age, but the decrease in CMA seems to aggravate the course. I show you an example of an age-related diseases, but we have shown them in others. So now, what can we do to prevent this. So something that we've been looking, and we have here before from Steve about caloric restriction, in collaboration with Rafa de Cabo, we wanted to know whether CMA changed during caloric restriction, because it's one of the most generic interventions against aging. And as you can see here, compared to the decrease that you see with age in animals in CMA, the ones that are caloric restricted in green, you can see that they preserve higher levels of basal CMA. We can reproduce the same if we use caloric restriction mimetics, those compounds that modify acetyl-CoA, and as you see in the images, you see a very nice increase on CMA. And then if we eliminate CMA and we gave these compounds, we lose the beneficial effect that caloric restriction and caloric mimetic have in many of these parameters of resistance to stress, suggesting that this upregulation of CMA that we see with caloric restriction might in part contribute to the beneficial effects. So in the last couple of slides, I just want to show you that we have also done some medicinal chemistry. So in collaboration with Ebris Gabathiotis, we develop small molecules that are able to activate CMA. They are they have very good biodistribution. So if you look, for example, in the liver on the top or the brain on the bottom, remember more dots means more active lysosomes, so you have very nice activity. We gave these compounds to different models of disease. So this is a neurodegeneration mouse model. It's the PS19, so it's a tauopathy model. And as you can see here, compared with the PS19 that has all this hypermotility and also lack of sense of risk, when you gave the compound that activates CMA for 3 months, you already have a very reduction of the behavioral phenotype, you improve memory, and if you compare, this is stainings for tau. So tau is the pathogenic protein, so on the top are the controls. In the middle are 3 regions in this mouse model of disease, and if we gave the compounds for 3 months, we can remove most of this pathology and this aberrant tau. We have used also these drugs in the context of retinal degeneration. So in this case, this is a model of retinitis pigmentosa, where the animals have very thinning of the retina and they lose sight. And what we find is that if we administer the compounds, so the compounds will be in green, the thickness of the retina is preserved, as you can see in the thickness with the arrow. So the thickness of your retina in the presence of the compounds is preserved because you don't have retinal ganglionar cells dead. And you also have less gliosis. The inflammation associated to the disease that you see there on green is decreased when we gave the compound. But of course, it's not only a beautiful retina. We also wanted to know if it's functional. And when we measure sight, these animals can see better because they have preserved retina. So in addition of models of disease, we have also tested them in other conditions. So for example, in models of physiological aging, and this is my 2 last slides. So Rabia gave these compounds to animals when they reach 18 months of age, and then we compare them how are they at 23 months of age. And when you look at different parameters, I'm showing memory. So memory is preserved in the animals where we gave the compounds. You also have better learning ability in contrast with the decrease that you see with age. Our aging animals with the compounds are learning better. And then if we induce seizures, remember we have a phenotype of hyperexcitability, so this increase in vulnerability to seizures that you see in elder individuals, in this case in elder mice, is completely eliminated when you restore normal levels of CMA. Maria, I wanted to convey, as I said, this is a pathway that decreases with age. The decrease phenocopy many aspects of aging and also accelerate the course of the disease. But I think we have a positive message because targeting of this pathway seems to restore or prevent many of the changes that happen with age, and also could have applications for age-related diseases. And I showed you a model of neurodegeneration, a model of retinal degeneration, but we have also done it in models of metabolic disorders and immune dysfunction. And of course, this is the most important slide. I only know about lysosomes, so any other thing is all done by the amazing people in my lab, people who passed through the years, and also the amazing collaborators that have been assisting in all these studies. This is the people who gave us the funding, and thank you so much for your attention, and I will be happy to take any question.
感谢 Professor Cuervo 的分享，请观众朋友们踊跃提问。
Thank you for the wonderful talk. I have a question about the CMA activation. So whether the effect of CMA activation can reduce cellular senescence or reverse the cellular senescence phenomenon, and whether if you stop using the compound, the rejuvenation effect can be maintained.
Yeah, so that's a great question. So we have Becca, one of our graduate students, her whole thesis is on the relation between senescence and CMA. And what we have found is that universally, when cells enter into senescence, they upregulate CMA. But when you do it in an old cell, if you get an old cell and you induce senescence, they do not upregulate CMA. And it's not necessary for establishment of senescence, because you still establish senescence in both cases, but the difference is for the reverse of the clearance of senescent cells. So if you don't have CMA, senescent cells cannot be identified by macrophages so well, and then they persist. So we think that lack of CMA contributes to the persistence of senescence. It's not that you are creating more, it's that you are not eliminating. And we have done both the drugs, but also genetic enhancement of CMA. And in both cases, you have a reduction of senescent cells in most of the organs, but you also improve the immune system. So it's a two-game, right? The senescent cell and the macrophage.
I understand the rationale in the tau model that you are probably accumulating these proteins when you don't have the CMA. In your wild-type animals that are CMA knockout without disease, you see this decrease. Do you see accumulation of unfolded proteins or UPR response? Can you rescue if you eliminate the UPR response? Is this accumulation of abnormal proteins is with driving the phenotypes?
Yeah, so sorry, I went very fast through that, but that's a very interesting question, because when we did the first model that we did was in liver, hepatocytes. And I told the student, oh, we are going to have a beautiful paper, you are going to take out CMA, there are going to be aggregates, oxidized proteins, it's going to be a mess. And nothing happened. I mean, they still have a phenotype, but there was not aggregation. So it turns out that in some organs, like the liver, you block CMA, and you have a compensatory increase of the proteasome and of macroautophagy. So you are compensating. The problem is that those pathways are activated at different times, so you lose the regulatory function. You don't have garbage, but you cannot terminate proteins at the time that you should to, for example, control glycolysis. The other interesting thing is that happens, for example, in fibroblasts, in hepatocytes, macrophages. When you go, for example, to retina, neurons, we eliminate CMA, and we don't see this compensatory activation, and we don't know why. So we've been trying to characterize how does the liver compensate to see if we can do it in the brain. And then the other sad news is that this compensation mechanism, even in the liver, it happens when you block CMA in a young animal. If you block later in life, you don't have this upregulation. So basically, you lose the ability to compensate even in an organ that was compensating. And then you start seeing aggregated UPR, you have the whole stress response because you are accumulating proteotoxicity.